Hello everyone, and welcome to this Gen Keynote webinar hosted by Jen. Thank you so much for joining us. Our subject today is integrated preclinical solutions to guide clinical development of bispecific antibodies. Today's webinar is sponsored by Crown Bioscience. I'm Uduak Thomas, Senior Editor with Jen, and I'm delighted to be your host for today's webinar. We have a superb speaker lined up for you. Dr. Rajendra Kumari is the Executive Director for Integrated Solutions at Crown Bioscience, and she will be the main presenter for today's topic. And then joining us for the Q&A part of the webinar is Dr. Henry Lee, Chief Executive Officer and Chief Scientific Officer at Hanks Biopharmaceuticals. Now, before I hand off to Dr. Kumari, let's go over some background on the topic of the day. Novel therapeutic modalities such as bispecific antibodies that simultaneously target two antigens are demonstrating promising clinical results in oncology. Some of the benefits of this type of therapy include enhanced specificity, reduced side effects, and increased efficacy. Despite the challenges in developing bispecific antibodies, progress in protein and antibody engineering has resulted in an increase in bispecific antibodies and their preclinical assessment. In this webinar, you'll learn about recent advances in preclinical tools, translational research, and the use of omics data that are narrowing the gap between the lab and the patient. You'll also hear about a novel bispecific antibody that targets both PD-1 and CD47, as well as some of the challenges and opportunities for an IO strategy to target and modulate the adaptive and innate immune system. Lastly, our speaker will offer some evidence-based identification of potential clinical indications. All right, without further ado, let's get started. Dr. Kumari, over to you.
Thank you for joining today's webinar. My name is Rajendra Kumari, and I will be presenting on integrated preclinical solutions to guide clinical development of bispecific antibodies. And this is a joint presentation with Dr. Henry Lee from Hanks Bio, who will be joining me to take any questions at the end of the presentation. So I will start by briefly overviewing bispecifics in oncology and then discuss some of the preclinical research tools and solutions to apply to screening bispecifics by giving a case study that will describe the path to IND for Hanks's novel bispecific antibody HX009. So tumor antigens are antigenic substances produced by tumor cells that can trigger an immune response in the host. And these can be tumor-specific antigens or tumor-associated antigens. And in most cases, they are found on the tumor, but in some cases, they can also be found on normal cells. So they are useful tumor markers in identifying tumor cells with diagnostic tests, and also are potential candidates for cancer therapy to increase tumor immunity. And these therapies, which are shown here, can include CAR T cells and antibodies, which recognize these antigen binding sites. So antibodies can include the sort of canonical IgGs. There's also modified antibodies such as ADCs, bispecifics. There's also bispecific T-cell engagers and even trispecific antibodies. And then you have the fragments which retain certain antibody functional features. But there are limitations to monoclonal antibodies in cancer, and this includes low tumor penetration and retention, and this results in poor target engagement. Or there may be good target engagement, but failure to induce a sufficient immune response. And this can include T-cell activation. And these modifications shown here to these antibodies aim to overcome some of these limitations. So, for example, for bispecifics, these actually present a range of opportunities to improve efficiency as well as efficacy, as they are usually designed to simultaneously bind to two antigens. And this can improve the selectivity and exert that sort of biological and pharmacological effect in the tumor area rather than in a systemic immune modulation, which then also minimizes the potential side effects and systemic immune-related adverse effects. So as two functional pathways are modulated, there may be greater chance of avoiding resistance to treatment in some cases. Now, and alternatively, a bispecific can be designed to engage a tumor cell receptor and a cytotoxic immune cell, and this results in immune cell recruitment. And this T cell engaging capability enables targeted or specific cytotoxic immune cell activation and promotes better clearance of those tumor cells. In addition, bispecifics can be potentially cost effective over something like a two antibody combination therapy because there's only one antibody being used as opposed to two to target two different targets. So progress in the field of antibody or protein engineering and recombinant DNA technology really has led to the establishment of different platforms to generate a variety of bispecific antibodies. And the standard antibody structure, which is shown here on the left, can actually be altered with different configurations that are shown here. And for example, used to generate different bispecific antibodies, which is nicely reviewed in this paper. Now in oncology, there's two bispecific antibodies that were approved for clinical treatment. The first, catumaximab, targets CD3 and EpCAM. And this was approved by the EMA in 2009 for intraperitoneal treatment of malignant ascites. Now, Amgen's blinatumomab is a bispecific T-cell engager targeting CD3 and CD19. And this was approved by the FDA in 2014 for the treatment of Philadelphia chromosome negative B-cell acute lymphoblastic leukemia, and first to be approved actually by the FDA to treat leukemia cells through human T cells. Now in clinical trials, there are approximately 60 different bispecific antibodies targeting both solid tumors and hematological tumors. And the majority are targeting the engagement of immune cells as shown in the blue section. There's approximately about a quarter that are blocking signaling and then a small portion that are delivering a payload. And then the next generation of bispecifics that are emerging are summarized in the table. And these have been rationally designed in some cases to overcome some of the specific challenges that have already been observed. For example, the development of multi-specific antibodies and targeting neoantigens to overcome tumor heterogeneity, as well as mutational burden, or to avoid some of those side effects and cytokine release syndrome that is typically observed. And as the number of bispecific antibodies increases, preclinical investigations also need to accommodate and interrogate these new targets and therapeutic approaches. And to assess these types of immunotherapies, there really isn't a single model or perfect preclinical model that can mirror the patient. So therefore, an integrated preclinical approach is needed or adopted to evaluate the phenotypic features and measure efficacy and toxicity in the most patient-relevant system. So this may involve actually multiple models and tests to assess certain or specific aspects of the drug's anti-tumor activity to build a complete picture. And as an example, an overview of some of those non-clinical solutions is represented here, but the appropriate solution will actually be determined by the targets or the targets for bispecifics, the therapeutic modality, such as large molecules, as well as the disease models. And there's many new technologies and innovations occurring that can potentially provide further insights or unique insights into the therapeutic potential of bispecifics, and we'll touch on some of those shortly. So when it comes to evaluating some of these strategies in preclinical models, it can be difficult knowing which models can be used to answer such questions, either in vitro or in vivo. And there's a lot of options and new technologies, like I mentioned, that have emerged to improve the predictive and patient relevance, and that the original historical models, which were seen as being poorly predictive, which is shown on the left-hand side. So you can see the emergence of new models as you move to the right. So, for example, there's now biobanks of patient-derived models created over the last few decades, and these can be PDX models, the patient-derived xenografts, these can be the organoids, and they're able to better predict clinical response. And combined with next-generation sequencing and AI, there can be improvements in patient stratification, improvements in biomarker identification, and predictive modeling. And also in vitro, the advances in culturing, assay conditions, and data acquisition for that sort of complex in vitro 3D biology that's emerging has really provided more valuable insights early in drug discovery pipeline, as well as potentially screening patient tissue directly, either ex vivo or in 3D organoids in vitro. And this again, combined with the technology such as high content imaging and AI-led analysis, is really making a big difference. There's also the emergence of new immunotherapeutics, so the demand for immune oncology relevant models has increased, and again, engineering technology like CRISPR modifications have allowed new models to be made to improve the immunodeficiency of mice, to allow better engraftment, to create humanized models. There's also knock-in, knockout, and mutational modifications that are made to really develop the right model for the therapeutic target, which may not have been available in the past. And also then you have animal imaging, which has also improved, and that now enables more complex biology and later stages of disease to be modeled and evaluated with much higher accuracy and throughput. Now, some of these models shown here, I will describe in more detail, especially in the context of bispecific antibodies. But just want to mention that you can find a lot of the information on these models, the characterization, patient information, and also perform searches or genome protein searches via our databases. And these databases are listed in the bottom right-hand corner, and you can access them via our website. So for selecting the appropriate models, you really do need to understand the limitations and the advantages. And this table highlights both those benefits and shortcomings of the main sort of models and their potential for screening bispecific antibodies. And actually carefully selecting the preclinical model can really make the difference. So for example, xenograft models, these have many advantages, allow the study of human-specific targets. So this is essentially formed in a vacuum, as the interactions with other immune cells or healthy human tissue isn't existent. You then have humanized immune reconstituted models, and this again provides a mechanism to allow human target evaluation in both the tumor and also the human immune system, which has been engrafted into the animal. But in some cases, the lymphoid and the myeloid compartment varies depending on the type of humanization and the human donor that's used. You then have syngeneics, and the main advantage here is this model is in mice that are immune competent, and this allows for observations to be made of these bispecifics in the context of a functional or fully functional immune system with those immune targets. And additionally, in some cases, syngeneic models can reveal on-target off-tumor toxicities, as the healthy murine tissue can express some of those target antigens, sometimes in low levels, sometimes at levels that are relevant to the patient. However, the key disadvantage is that this is a mouse biology, so a mouse surrogate antibody is needed, and also the mouse biology doesn't always accurately replicate the human immune biology. Now, at the bottom, there's transgenic mice have, in some cases, overcome some of those shortfalls of the syngeneic models as they express the human antigen or target, and also have the potential for on-target, off-target effects in specific sort of antigen-expressing healthy tissues. So the transgenic do still employ the murine immune system and it's fully intact, like the syngeneics, but it does allow for the study of human antigens similar to the xenograft system. So as you can see, one model may not answer all the questions, and several may be needed to be employed based on those limitations and advantages. And I just wanted to discuss a little bit more at the top there, the organoid models, as this is a novel technology that is being applied for many therapeutics, including bispecific antibodies. Okay, so some of the key features are shown here, and this is actually in comparison to other in vitro systems like the standard 2D and 3D spheroids. And the key difference is that the organoids, which are derived from adult stem cells residing in that tumor and using the technology developed by the Cleavers lab, these organoids have self-organizing properties and are made up of a mixture of stem cells and differentiated cells. like those you can find in the corresponding organ or the tumor in vivo. And the structure actually resembles the microanatomy of the tissue from which they were derived from, and you don't see this in 2D and 3D spheroid cultures. In addition, organoids have the ability to be maintained in long-term culture, so they can be passaged and expanded and banked. And this is again due to the population of those stem cells, which can replenish the cells, the tumor cells, and this again cannot be achieved with spheroids. So the term organoid should only be used to describe the mini organs or tumors that are derived from stem cells. And as a result, this also has a huge advantage because it means that you can derive organoids from both normal tissue, but also organoids from the disease tissue, such as tumors. And these can be made directly from the patient and from the clinic. And these have a lot of applications. So they've been used for drug screening, co-culturing with other cells like immune cells, and also high content imaging can be applied as well as biomarker evaluation. Now, to find details of the different types of organoid models, so we have over 600 of them from 15 different cancer types. You can actually find these in our online database. And also you can refer to an earlier Gen webinar to learn more about their application. So in this study, which was published in Nature Cancer last year, we were able to demonstrate the value of high content imaging with organoid screening. And this actually led to a lead candidate being identified for clinical trials. So there was firstly a primary screen, and this involves several organoids models that were first screened with over 500 therapeutic antibody to narrow that selection down to a handful of candidates. And then the second screen used over 20 colorectal organoids, colorectal cancer organoids, to see the differential responses across that large cohort of patients that they represented. And in the end, to select a compound that showed very high selectivity towards the tumor organoids and not to the normal organoid, which were also included in the screen. And this was also done in comparison to a standard of care agent, cetuximab, that is in the clinic for use in patients for colorectal cancer. And the data here on the left, on the right, shows the internalization and the degradation of EGFR receptor by Meros's agent MCLA-158, which was more rapid in comparison to cetuximab and was mediated by LGR5. But this wasn't seen in normal cells, which lacked the LGR5. So this led to the identification of a single agent that could target EGFR positive and LGR5 positive colorectal tumors with limited toxicity to normal tissue. And so this drug, like mentioned, is now actually in clinical trial. And this actually demonstrates how fast and effective organoid screening can be for large molecules like Bispecific, and how the phenotypic readout by high content imaging can support that lead identification and that potential biomarker identification. So in the next section, I would like to do is provide a case study that uses the preclinical translational solutions to advance a bispecific clinical candidate, HX009, and this is a first-in-class bispecific antibody targeting PD-1 and CD47. It will go through the rational design, the configuration of the different assays that were used to guide on that clinical development. Okay, so as you know, PD-1 together with its ligands, PD-L1 and 2, are overexpressed which are overexpressed in tumors, are key adaptive immune checkpoint molecules, and these regulate T cell function, activity, and drive that anti-tumor mediated immunity. And there's been robust clinical validation demonstrating that this is the most effective immuno-oncology treatment target with a number of therapeutic antibodies already targeting either PD-1, such as pembrolizumab or nivolumab, or there's also anti-PD-L1 agents as well. So the CD47 is a don't eat me receptor. It's often overexpressed on cancers where CD47 high is associated with poor prognosis. And its ligand, SIRP-alpha, is actually found on the surface of phagocytic cells like the macrophages and the dendritic cells, and this blocks phagocytosis. And this CD47 SIRP-alpha interaction is critical to both innate and adaptive anti-tumor immunity. So for the adaptive immunity, this involves dendritic cell presentation to T cells. And then more recently, it was reported in melanoma that the binding of SIRP-alpha-2 expressed on the tumors with CD47 expressed on the CD8 positive T effector cells co-activates the T effector cells with anti-PD-1 and potentially sensitizing the PD-1 blockade and reversing T cell exhaustion. So both these immune checkpoint molecules are considered important cancer drug targets, with one approved and one to be approved. And to date, actually targeting CD47 has been achieved with an investigational anti-CD47 monoclonal antibody drug, magrolimab, and with a SIRP-alpha fusion protein. But the main sort of challenges that are seen with these agents in the clinic has been largely due to the sort of modest single agent anti-tumor activity, but also the association with a variety of dose limiting toxicities. And this is due to the fact that these anti-CD47 target therapies cause anemia and thrombocytopenia, which is attributed to the broad sort of CD47 expression on megakaryocytes and the erythrocytes. Now, like any antibody, bispecific antibodies can be modified in countless ways to customize its functionality, to enhance its efficacy, also to modulate the sort of immunogenicity and increase the half-life. So there's a number of sort of different strategies that are being employed to improve these anti-CD47 antibodies. And actually, in theory, you could look to design or take some of those unique features of these antibodies to hopefully overcome some of those toxicities. And this is something that has been considered as the sort of strategy for HX009. So like I said, this was designed to leverage and maybe synergize some of those limitations to achieve dual targeting of both the innate and adaptive checkpoints and create a novel biologic which has enhanced anti-tumor activity, but also reduced hemotoxicity. So firstly, HX009 was rationally designed using HX008 framework, so the original frame, which is a humanized IgG4 monoclonal antibody developed for the treatment of solid tumors. And this includes GI cancers, melanoma, liver, non-small cell lung cancer, and breast cancer. So this antibody, so this is pucotenlimab, it binds PD-1 and blocks its interaction with its ligand PD-L1, PD-L2, and thereby restores the ability of immune cells to target cancer cells across a wide range of tumor types, as mentioned. It's very stable biochemically, and it has highly favorable PK properties. It received a conditional first approval in China last year for the treatment of patients with unresectable metastatic microsatellite instability high tumors or mismatch repair deficient solid tumors. And this includes patients with colorectal cancer who've experienced disease progression after previous therapies such as 5FU, and also patients with advanced solid tumors that have also experienced disease progression. So a very highly successful agent that's come to market. And also last year, it was approved in China for the treatment of unresectable and metastatic melanomas who have failed previous systemic therapy. So the aim here was to ensure that HX009 maintained these desirable features of HX002, such that the sort of functional binding and blocking of PD-1 was retained, as well as that tumor targeting. So the extracellular CD47 binding domain of human SIRP-alpha protein is engrafted to the C-terminus of the heavy chain of HX008, as shown here. And the structure, so HX009 maintains that 2x2 symmetric format, and this ensures a stable structure that so the high affinity of PD-1 binding is maintained even with that SIRP-alpha domain attached. So this domain also has significantly reduced CD47 binding affinity, which aims to reduce the CD47 positive red blood cell binding and minimizes hematotoxicity. And this also minimizes the anti-CD47 sort of antigen sink effect that you would see, because it is broadly expressed on normal tissue as well. And so the idea that is that the anti-PD-1 binding will direct the bispecific via tumor targeting within that tumor microenvironment, helping or bridging that weakened SIRP-alpha binding to the CD47 on the tumors. So as shown on the right-hand side, that combined mechanism of action is with anti-PD-1 impacts adaptive immunity and also potentially reverses T effector exhaustion. And then on the left-hand side, the CD47 blockade suppressing the sort of don't eat me signals in innate immunity response, as well as impacting the adaptive immunity to promote antigen presentation to T cells, and also potentially reversing that T effector exhaustion. So actually, HX009 recently received investigational new drug approval to start clinical trials, and of this potentially first-in-class recombinant anti-CD47 PD-1 bispecific antibody. And this is a phase 1b/2 clinical study to be conducted in the US to evaluate HX009 in the treatment in patients with relapsed or refractory lymphoma who have failed standard therapy. And it's already been studied in Australia and China in both solid and tumors and lymphoma indications. So here the 2 phase 1 dose escalation studies, which was presented at ASCO in 2021, shows favorable toxicology and PK profiles, and also preliminary efficacy was observed, which includes multiple complete responses, partial responses, as well as stabilization of disease. And this is in both the solid tumors and the lymphoma. So the development of these novel new drugs, such as HX009, really does rely heavily on the use of experimental models to quantitate the anti-tumor effect, the specificity and the advantages in the most sort of relevant biological context as possible. So in this case study, we described that integrated preclinical testing to confirm that design properties and the anti-tumor activity. And this was also jointly published in scientific reports as shown here. So I'll share some of the data that's been presented. Okay, so if we start with interrogating the design properties using different in vitro systems, so the first step was to examine the binding to the respective targets. And here the binding as measured by ELISA confirms the interaction of HX009 to recombinant PD-1 and CD47 proteins. And in comparison to the parental HX008, you can see that the anti-PD-1 binding was identical with the same sort of EC50. So the binding affinity wasn't reduced in any way with HX009. The EC50 for binding of CD47 was approximately 50 times less potent than the PD-1 binding, as well as lower than binding affinity for other similar sort of agents. But overall, the aim was to show that there was lower binding, and that's what has been achieved. And the data at the bottom shows a competitive ELISA assay to quantitate the blocking potential of HX009 against its target receptors. And their sort of respective ligands. And again, this is using recombinant ligands for PDL1, PDL2, and also SIRP-alpha. And you can see that the PDL1 blocking dose response curve is only slightly lower than that for HX008, while the PDL binding blocking dose response curve is slightly different, but overall there's very similar sort of IC50, sorry. And the SIRP-alpha Fc blocking IC50 of HX009 is shown to be approximately 23 nanomolar on the bottom right-hand side. And this is in contrast to blocking dose response curve for the native SIRP-alpha Fc with an IC50 of approximately 1.4. As well as margolimab with an IC50 of 1 nanomolar. So again, demonstrating very low binding affinity feature of this bispecific antibody for CD47. So HX009 binding to PD-1 to positive cell lines, receptor positive cell lines, was also assessed using an engineered 290 cell line and assessed by flow cytometry. And the dose response curve here demonstrates that HX009 specifically binds to the PD-1 overexpressing cell line with an IC50 of approximately 2.9 nanomolar. So again, very, very selective and high affinity binding. And also the HX009 binding to CD47 positive lymphoma cells. So this is the RGB and the JERCAT T lymphoma was high in contrast to the minimal binding that's seen with HX008. So again, confirming that there is dual binding that's occurring. And then the next step was to determine the functional impact of the respective signaling downstream, which would be actually critical for CD47 interaction, given that there is a weaker binding affinity. And this was evaluated using a receptor cell line system, a reporter cell line system, where the reporter readouts on luciferase activity represents a surrogate function readout of a specific ligand or receptor interaction. So here the JERCAT cells were engineered to express IL-2 promoter-driven luciferase reporter, and then co-incubated with CHO cells expressing both TCR stimulatory molecules and PD-L1. And this luciferase readout represents or mimics the T cell activation by any sort of PD-1, PD-L1 blockade. And what's interesting is that the HX009 demonstrated consistently higher functional readouts, nearly 3 times more potent than HX008. And if this is true, then it's possible that the CD47 engagement in T cells could potentially enhance the T cell activation pathway by PD-1 blockade. And this, as has been mentioned, was shown earlier. So therefore, HX009 could be more effective than immune checkpoint inhibitors such as PD-1 alone. But all of these would need to be further sort of qualified. Similarly, we also assessed the CD47 function blockade by HX009 using another cell reporter assay where the JERCAT cell line was stably transfected with chimeric SIRP-alpha receptor composed of a CD47 binding domain of SIRP-alpha, the transmembrane domain of CD28, and then the intracellular CD3 epsilon signaling transduced domain. So the CD47 gene was actually removed from here, so to avoid sort of self-binding with the cell. So when CD47 binds, the cell triggers a strong CD3 activation, which leads to cell death, and this can be prevented by anti-CD47 antibody binding or soluble SIRP-alpha mediated neutralization. And this was able, and so in this assay, we were able to confirm the functional blockade of CD47 by HX009, as shown here on the bottom right. And clearly, as you can see, with lower potency as compared to margolimab and SIRP-alpha FC control. Again, using the reporter cell line, the combination of HX008 with anti-SIRP-alpha was evaluated in comparison to HX009. But despite the sort of high affinity of the anti-SIRP-alpha, this didn't actually enhance HX008 as seen with HX009 alone. And this suggests that the cis binding was driven by that PD-1 binding, which is, a key feature of this novel bispecific. And then to address the sort of functional aspects of T cell activation by PD-1 blockade, this was assessed by 2 very common assays in vitro. So the first, which is shown here, is exposure of SEB pre-treated PBMCs from 2 donors to HX009. And as a super antigen, SEB activates those T cells at low concentration and actually for a long time, and this renders them exhausted. And so what's shown here is that the exhausted T cells can be reactivated by the anti-PD-1 antibody and produce interleukin-2 and interferogamma that can be then measured as readouts for anti-PD-1 functions. And in both donors, so this is the 2 donors shown here, can activate, HX009 can actually activate or reactivate the exhausted T cells, similar to HX008. And this again confirms that the PD-1 functional capability and blockade by HX009 has been retained. Second, a standard mixed lymphocyte reaction assay was used to assess HX009's ability to enhance T cell activation during the mixed lymphocyte culture by directly measuring again interleukin-2 and interferon gamma production. And as shown here, treatment further enhanced the production of both cytokines in a concentration-dependent fashion, similar to that of HX008. And again, further confirming that the functionality of this antibody was retained. And also to, most importantly, demonstrate how the binding of HX009 compared to other CD4 binding agents to red blood cells. So this was assessed by FACS, and as you can see here on the left, HX009's percentage binding, or the sort of mean fluorescence intensity, was very low. So that's in red in comparison to margolimab in green. And this was also the case for platelet binding at the top here. And also in the CD16A binding assay that can be used to monitor binding of IgG-Fc regions to the human CD16A receptor. This is obviously typically to select the most potent antibody for use in ADCs. Here showing the opposite and the lack of binding of HX009. So this therefore supports the sort of design rationale for avoiding drug-induced thrombocytopenia and anemia, which is observed in many of those anti-CD4 targeting therapeutics. So now we looked at investigating anti-tumor activity in vivo models that specifically can model that immune oncology tumor microenvironment. And so there's many models that can be used here, but the ones that we've in the table here are shown in the paper, and so I'll describe those in a bit more. So there's not, like I mentioned, one perfect model for in vivo models for this type of therapeutic, but hopefully by building a picture using different models, a better understanding of the efficacy can be visualized. So firstly, cell-derived xenograft models, which are extremely valuable for early drug discovery, provide insight into that drug sensitivity, the cell biology, and the signaling pathways. And there's actually hundreds of cell lines to choose from, and we've got over 250 well-characterized models and validated in different sites in mice, and the data and information on these models can be found in our database called Xenobase. You can also use a database to search for specific markers, such as expression of CD47, using the sort of gene search tool in that database. So RARGB and Corpus 299 have good CD47 expression, just shown here on the left. And actually the NOD strains which are used here have mutations at its SERP alpha. So cross-recognize human CD47 in these lymphomas, and the principal basis actually for testing biologics targeting human CD47 using xenograft tumor models really does revolve around the fact that these polymorphic SERP alpha and the NOD SCID mice interact with the human CD47, and that then in turn supports the growth of these xenografts. So in the first study, subcutaneous Raji xenograft treated with HX009 at 10 mg per kg induced significant growth delay of approximately 54% TGI. And this was actually similar to SERP alpha FC treatment as well. And again, confirming the sort of anti-lymphoma effects, which can be attributed to the CD47 directed biological or unblocking of the don't eat me signals seen in this model. Now, of course, this model is devoid of functional human lymphoid immune system, so there isn't any contribution from the anti-PD-1 component of HX009. And to also note that the observed anti-lymphoma activity in this xenograft study was direct targeting tumor cells for by CD47 in the HX009, and this resulted in a significant anti-lymphoma effect despite the sort of weak binding of this particular molecule for CD47. And in the second study, Corpus 299, which is T-cell lymphoma, this was co-transplanted with human PBMCs with the intent to measure the PD-1 blockade effect beyond just the CD47 blockade in this Raji model. So this PBMC humanized mouse model, which is called Mizeno, which is described here, is used to test a range of different immunotherapeutics directed at different specific targets. So just a summary of those in that table. And also you can learn more about humanized models in the publication. So HX009 induced remission in a dose-dependent fashion in this model. And actually with statistical significance to the high doses. But HX008 showed little anti-tumor effect, which you would expect, obviously due to the lack of sort of autologous anti-tumor immune system in this model, and specific T-cell engagement. Again, confirming that the observed response of HX009 was really mediated by the anti-CD47, which is encouraging because, like I said, this has a weakened binding affinity. And given the absence of the sort of competent autologous immune system in these conventional xenograft models, including limited amounts in the Mezino humanized mouse model, you can't really effectively assess the sort of PD-1 blocking effects of HX009. So the alternative type of humanization is to substitute mouse gene targets by human counterparts, which is achieved by genetic engineering. So this is knock-in both in syngeneic tumor cells, so these are called HU cells, and in the mice, which are HU cell, and this creates a new humanized mouse model. So the HuGEM is an immune-competent chimeric mouse model engineered to express those human drug targets, such as those checkpoint targets. And the HU cell is the mouse tumor cell that has been engineered to express the humanized ligands. And these models, therefore, are employed to better understand the sort of anti-tumor effects of biological therapeutic agents, which are intended for human use. So there's no mouse sort of surrogate here. And there's numerous different types of targets that can be knocked in, which is shown or summarized in that table. You can also have human CD3 HuGEM models, which with sort of traditional syngeneic models can be used to evaluate CD3 targeted bispecific or T-cell engager antibodies. And again, we have a lot of data to demonstrate how these can be used. For today's case study, the model that's going to be described or used is a quadruple knock-in PD-1, PD-L1, CD47, and SERP-alpha HuGEM in BsAbs mice. And this was constructed so the strategy for construction is shown below. And the human CD4 knock-in syngeneic lines that were used are 820 B-cell lymphoma or the MC38 syngeneic line. So this humanized syngeneic mouse B-cell B lymphoma A20 model contains the autologous sort of tumor immunity that can be employed to assess that dual targeting mechanism of action sort of simultaneously. And the results demonstrated that while the single agents targeting either PD-1 via HX008 or CD47 via SERP-alpha FC do show anti-tumor activity, the dual targeting by HX009 induces synergistic anti-tumor activity. And these data in this complex model system leverages the need for the host innate immunity and strongly supports the notion of enhanced treatment benefits through that sort of dual targeting of both CD47 and PD-1. So recently, that's shown here, and then recently, as mentioned, a new mechanism of anti-PD-1 mediated T-cell activation by SERP-alpha on tumor cells interacting with CD47 on the CD8 positive T effector cells in the tumor microenvironment has been described, and it is possible that the observed anti-lymphoma activity from both the anti-CD47 function of the HX009 could be via the HX009 SERP-alpha interaction either with the tumor cell CD47 or with the T effector CD47 or both. In the CD47 humanized syngeneic mouse MC38 colon cancer model, which was also implanted in that quadruple humanized mouse, the results showed that the single agent CD47 blockade had little activity, so that's the yellow curve, whereas the PD blockade had significant activity. And overall, HX009 slightly had a higher increase in activity. And this potentially is due to the lack of CD47 component in the system. To sort of further test this, the CD47 humanized MC30 cell line was also then used in a humanized PD-1 HuGEM model as shown here. in this sort of humanized system, the single agent CD47 blockade, again by SERP alpha FC, showed little activity. So again, that's that yellow curve. And then in this system, we also tested Keytruda just to kind of benchmark it against the HX009 activity. And Keytruda, as expected, showed anti-tumor activity, which is the orange curve. And we did actually look at it in combination with SERP alpha FC to see if there was any further additive or synergistic response, and it wasn't. And the HX009 had similar activity to Keytruda. So this again confirms the anti-PD-1 blockade of HX009 in the absence of any CD47 blockade, and also that there are some tumors where one or the other target may not be expressed, but still these agents are superior because of that dual targeting, despite the fact one mechanism cannot be blocked. So if we look further at the potential indication selection, so we showed that the lymphomas had good response in the CDX. So we want to kind of explore this further in PDX models. So lymphomas are a heterogeneous group of malignancies. They're comprised of both highly aggressive and indolent diseases. And there's, many different, over 80 different subtypes of non-Hodgkin's lymphoma. And with the most commonly used chemotherapeutics such as CHOP and R-CHOP for non-Hodgkin's lymphoma. Now, the emergence of biological therapies such as rituximab targeting CD20 really has transformed the sort of management of this complex disease. And now there's actually a wide spectrum of agents used to treat these diseases, including the monoclonal antibodies, ADCs, and CAR-Ts. And in the case of lymphoma, the approval of PD-1 targeted drugs, including pembrolizumab and nivolumab, for the treatment of Hodgkin's lymphoma and nasal NKT cell lymphomas has represented or shown good improvements in this disease, but the activity of those checkpoint inhibitors in other diseases such as non-Hodgkin's lymphoma has been limited. And currently, CD47 SERP alpha blockade with the megolimab and SERP alpha fusion protein have yielded encouraging clinical activity, but again the challenge is those sort of modest monotherapy activity as well as the dose limiting toxicity. So the theory in designing the sort of feature or using the features of the CD47 targeted therapies could circumvent some of those toxicities and leverage, and what we're hoping to do is leverage some of that with the HX009. So what we did was to look at our PDX collection. So we have over 2,500 PDX models, which are can be searched for in our database called HuBase, looking for lymphoma models to identify potential ones that could be used in a mouse clinical trial. So the working hypothesis, so could those immune checkpoints high expression be a predictor for response? So is it actually a reasonable to speculate that the expression levels of these HX009 targets in lymphoma could be important parameters that influence the efficacy? So we first needed to kind of know if the lymphoma PDXs express these immune checkpoints. So we investigated the sort of molecular epidemiology of these checkpoints in a cohort of PDXs, either mRNA levels by RNA-seq or protein levels by IHC. And here the correlation of CD47, PD-L1, PD-L2, mRNA levels among the diffuse large B-cell lymphoma PDXs was evaluated, and as you can see, the corresponding R values or P values, which are displayed, shows actually what's most interesting is that the three markers PD-L1, PD-L2, and CD47 are actually co-regulated among the cohort of lymphomas that were evaluated with actual statistical significance for those PD-1 ligands and the CD47. So that was quite a significant sort of discovery to kind of identify here. A similar trend of the sort of co-regulation has been seen in DLBCL patients, although the correlation was short of similar sort of statistical significance, and that possibly is due to the sort of smaller sample size. But again, this does actually further support the rationale for dual PD-1 CD47 targeting for treatment of lymphoma. And here the representative images of lymphoma PDX for IHC staining for CD47 and PDL1 demonstrates that there is actually varying degree of, for example, CD47 protein expression. And then the expression levels of both CD47 and PDL1 in those PDX, as well as the IHC scoring, is summarized in that table, so you can see sort of side by side what that looks like, and how they correlate, and again at the bottom, the correlation of PDL1 and CD47 H scoring shows or seems to suggest that there is some co-expression of those immune checkpoints. However, to note that the H scores of IHC are not well correlated with mRNA levels of these models by RNA-seq for CD47, and that's shown on the left-hand side. In contrast, you can use a commercially available companion diagnostic kit, and that does correlate with mRNA levels by RNA-seq for PDL1 with statistical significance against the IHC score. And also just to highlight that here, an IHC or RNA-seq assay could be potentially developed as a predictive biomarker for HX009 treatment if there is robust correlation between the target expression and the pharmacological efficacy. Now, especially as already there is a PDL1 IHC companion diagnostic available, and this is commonplace in the clinic for PDL1 treatments. So having something similar for CD47 will be highly favorable for clinical development, and specifically for HX009 patient selection. And then to validate the working hypothesis that the expression levels of immune checkpoint could predict HX009 efficacy in lymphoma, we tested this in that small cohort of lymphoma PDX in a mouse clinical trial format. And then, so the data is shown here, and obviously due to the immune deficient nature of the PDX models, only CD47 targeting could be assessed as above, as described above for the xenograft modeling, because of the sort of immune deficient nature of those animals. We did actually include CHOP treatment just as a standard of care and a positive control, and the tumor growth curve inhibitions are shown here, and also these are summarized in the table alongside those mRNA and H-score data. So the CD47 high models may represent better responders to HX009, or CD47 expression could be a positive predictor for the efficacy, and the sort of high levels are kind of highlighted in red here. So although we could not demonstrate the influence of PDL1, L2 levels in HX009 pharmacology in these PDX models due to that immune deficiency, it's still reasonable to speculate that the high expression of these ligands could potentially also be positive predictors for efficacy in humans due to the co-regulation that we reported, and also the dual targeting of HX009, and therefore should likely be more effective than the respective single targeting in human lymphoma patients. So to summarize, so the objective was to validate the rational design of HX009 bispecific antibodies as a dual innate adaptive immune checkpoint inhibitor with the reduced affinity with CD47 and the preliminary validation of HX009 as a lymphoma treatment. And this was achieved through the in vitro characterization confirming the bispecific's dual binding with the sort of desired affinity and functions, with in vivo xenograft lymphomas confirming CD47 blocking mediated anti-lymphoma activity despite that reduced binding. And then the in vivo humanized syngeneic lymphoma HUGEN modeling also confirmed the sort of superior anti-lymphoma activity because of that dual blocking over single immune checkpoint blocking. And then the mouse clinical trial using the PDX models with differential CD47 expression showed that CD47 high models were more likely to respond to HX009. And this also leads to that the phase 1/2 study is ongoing in patients with relapsed refractory lymphoma, and including both the B and T cell subtypes, and has shown encouraging preclinical anti-lymphoma activity. And also there is potential for developing a CD47 IHC companion diagnostic, which could basically hopefully guide on patient stratification in the future. So with that, I'd like to thank you for your attention. If you have any further questions, I'm happy to take those. And also you can find further resources and information on our website, as also access to the databases. Thank you so much.
Thank you very much, Dr. Kumari, for that fascinating talk. Now, in a few moments, we'll move into the Q&A session for the webinar, and so we really hope you'll stick around for that. Before the Q&A session, a quick reminder that this webinar will be available to view on demand in the next 24 to 48 hours. So please keep an eye out for that, and do share the link with your colleagues who might not have been able to tune in today. Thank you, Dr. Kumari and Dr. Lee, for the wonderful presentation. We're now going to move into our Q&A session. But before I do that, I just want to remind everyone that this webinar will be available to view on demand in the next 24 to 48 hours. So it would be wonderful if you could share the link with your colleagues or anyone you know who might not have been able to tune in today. All right, it's time for our questions. We have some really great ones here for our panelists, and I think this is going to be a really rich and interesting conversation. So my first question is for you, Henry. What other indication did you consider? Are there any benefits for solid tumors?
Yes, that's definitely the thought. The objective of designing this bispecific antibody is really geared for a PD-1 plus or super PD-1. That's the idea, right? So PD-1 has been great success. So the idea is to go to the indication area that has not been well served by the PD-1 antibody. Obviously, solid tumor some of the solid tumor are including into those indications we're considering. For instance, TNBC recently I think had PD-1 approval quite recently, but a lot of solid tumor, for example, pancreatic cancer, gallbladder carcinoma, and some subtype of melanoma. Those are the some of the example of the solid tumor we're trying to test in HX009. Yeah, and just to add a little comment in particular. For example, there was a report late last year, really the cancer, I think it's Cancer Cell report that they found out that some of melanoma, which is the first approved for IO therapy, actually does not respond into PD-1 or CTLA-4. And later on, they found out that in that described in that report is because of the low expression of the SERPA. So they come up with a theory that actually CD47 and PD-1 dual targeting are actually required for the long-term reverse of the T effector cell exhaustion. So our idea is testing in those melanoma that of our bispecific antibody, which do provide this dual targeting. Okay, so that's basically the idea. But of course, I do want to add a comment that clinical development is another beast. It's not something we have demonstrated working in a translational area is not necessary. We're successful in clinical. In clinic, there's too many other consideration. In certain indication, especially after several lines of the treatment, can you recruit sufficient patient? Or those patients will be able to go through the further trial. All right. The combo therapy usually are required because there is this SOC. You have to use ethically. And then that means you're going to need multiple group, and you have to randomize it. So the trial become very big, and clinical trial are very expensive. So all those things will contribute to the complexity of translates your translational readout into the clinical testing. So I just want to emphasize that. Yeah.
Yeah, absolutely. Thank you very much. And I'd actually like to stay with you, Henry, for this next question. Did you try a CD34 positive humanized mouse model for any of the xenografts?
We have not. Yeah. So that's probably just due to the economical reason. We just have not yet. Yeah. It could potentially be tested.
Yeah, absolutely. And then, Jen, I'm going to come over to you for this question. Would an organoid HX009 trial provide similar results but faster?
I mean, for indications like lymphoma, this would be challenging as the tumor organoid biobank is limited to solid tumors, typically of epithelial origin. So yes, they're not for hematological diseases. But for other indications, you would also then need to co-culture with immune cells, as HX009 is actually targeting immune checkpoints. So it does actually become a bit more complex. I mean, I gave an example in the talk of a Bispecific lead identification study, which was a great example of using organoids in a faster throughput. But those were targeting tumor antigens specifically and not necessarily needing immune cell compartment. But we have actually used co-cultured organoids for other immune oncology target evaluations and for bispecifics. So yes, it could be used, but just not as a high throughput.
Okay. Excellent. Thank you. Henry, I'm going to come back to you for this next question. Do you have any specific idea why the CD47 blockade in the MC38 with huge models was not observed with the SIRPA? SIRPA?
Yeah. I think there's probably many reasons which we do not really fully understand. The CD47 now actually by itself has a pretty bad reputation as a target because used to be think of as such a hot checkpoint like PD-1, but it didn't turn out to be that way. It is none of the CD47 targeting has been approved so far, particularly in the solid tumor situation. Very few actually solid tumor responded to CD47 blocking by itself. So that's pretty most people accepted. So that's why most of CD47 related trial has been conducted in the hematological malignancy area. So this is not a surprise. So in order to interpret the ineffectiveness, wonderful theory is that actually in order to really fully blocking the so-called don't eat me innate immunity checkpoint is there is a several parallel pathway in those controlling the don't eat me mechanism. So simply blocking CD47 just insufficient. Okay, so probably dual blocking or more than dual blocking are required, and this is one of the area had intensive research ongoing at this moment.
Okay, excellent. Still staying with you again, Henry, for this next question. Were any of the sensitive PDX also high MSI? And so this is a two-part question, actually. Was HX008 tested alongside some of the lymphoma PDX?
Yeah, first of all, I probably just comment on the lymphoma. Lymphoma trial or in a PDX panel. We did try, in some cases, tried HX008. So this is the second question. In there, obviously, we have not seen any effect. which is understandable because there's no T cell and there's no PD-1. So that's basically just consistent with what this model supposed to behave. With regarding to actually, be honest with you, I not too for MSI, the so-called sensitive, usually is referring to PD-1 treatment, and not to CD47 treatment. And when we talk about a PDX, which is xenograft, there's no PD-1, there's no T cell involvement. So that's not what we usually discussed. Yeah. Yeah.
Okay.
Not in the same context.
Perfect. Thank you. And so this next couple of questions I have are for you, Jen. So the first one I have is, can you co-culture organoids with PBMCs to monitor the ICI response?
Yeah, yes, absolutely. I think, yeah, just kind of expanding a little bit more on that last answer. So yes, organoid co-cultures can be used to evaluate immune checkpoint inhibitors, but obviously there is it is very much dependent on the combination of that specific organoid model with the immune cell donor. So ideally this should be an autologous system to measure that immune checkpoint inhibitor specific response. And then the other advantage of using an autologous system is that you avoid the sort of donor to donor variation in the non-autologous systems. But I mean, as you kind of gathered, that immune checkpoint inhibitors, although they've seen some good success in the clinic, there's only a small proportion of patients that actually respond to immune checkpoint inhibitor sensitivity. And we've actually seen that in some of our patient-derived systems, especially the ex vivo patient platform, which actually retains endogenous immune cells. And so that is also another alternative assay to the sort of organoid co-cultures. And in some cases, you know, the immune effector cells do need to be pre-activated in some of these autologous systems with a superantigen, and that is just because they may lack some of that tumor reactive T cells, which sometimes isn't always present in those patient PBMCs. But on the flip side, not all models have matched autologous PBMCs to use, so we do have to then defer to using the PBMCs from healthy donors. And of course, in some cases, that may lead to an alloreactive response, which can actually vary from donor to donor. So this may impact the ability to monitor a tumor a tumor antigen specific immune cell response. So it is challenging either way, but it is possible. Or, like I said, the alternative could be to use an ex vivo patient tissue assay.
Thank you, Jen. That was a great response. And so my second question for you is, what models are there for T cell engager bispecific antibodies that target CD3E?
Yeah, I mean, that's another good question. And yes, the problem has been the sort of poor cross-reactivity with mouse CD3. So standard sort of syngeneic and homograph models are not really ideal for human antibodies targeting CD3. So the humanized system is actually needed. And for CD3 or even CD3 EDG, you can use huge-M models which have fully humanized CD3E or the CD3 EDG targets on those T cells. And so these are very suitable for T cell engager studies. And these models have been characterized, so there has been shown that the T and the B and the NK cell development is quite normal, and there's also normal T cell activation, and there's different strains, so you can use these in BsAbs or C57 mouse backgrounds. So there's quite a large number of models available, and you can use different cell lines for these models as well. So and we can also look at the organoids for T cell engagers. So again, just co-culturing with the PBMCs. And actually, I didn't get a chance to kind of expand on this in the webinar today, but we do see specific tumor organoid killing that's redirected by those bispecific T cell engagers. And this can again show very tumor-specific antigen-mediated response, and that will be into a control antibody. And you can use models with maybe high or low antigen expression on those organoids. Again, this will kind of give you a better idea of whether that is a specific response. And these are very quick assays. They take about 48 hours of co-culturing, so you get a good readout very fast.
Excellent, thank you. I'm going back to you, Henry, for this next question. What toxicity testing has been done, and how is this correlating with the clinical data so far?
Yes. First of all, I want to make a comment. HX009 bispecific antibody was designed to reduce the toxicity have been seen, for example, in CD47 targeting, which is a hemotoxicity like platelet, red blood cell reduction, those type of hemotoxicity. And so basically, because of the design, those toxicity was really minimized. But even so, any pharmaceutical, you need to go through standard toxicology study. Before the injecting the human, you go through the GLP type of in this case, because it's a biological, you have to do like non-human primate as a tox species to test this. As I said earlier, because of the specific design, the toxicity are very, very little in the right range. And this compound has been tested in more than 70 people already. And the toxicity is all minimal. And it is correlated with the non-human primate data. And the actual efficacy data is much lower. For example, this compound we already dosed to human at 50 milligram per kilo. We did not see DLT, which is called dose limiting toxicity. And but however, efficacious dose is at much lower in probably 3. PD-1 is a 3 mg, right? So in 3, 5, 7, we also efficacy. So definitely it has a broad therapeutic window. Tox is not a major concern for this compound. Yeah.
All right. I have a few more questions, one for each of you. So Henry, I'll start with you. So this is a smart design. Could HX009 be used to treat patients who've relapsed from pembrolizumab? Excuse me.
Yeah. The idea or original objective is to create so-called PD-1 plus or super PD-1 that, for example, can deal with tumor resistant to PD-1. For example, some of those so-called cold tumor. Yeah, they definitely is for that purpose. And of course, can we significantly demonstrate this capability in the clinic setting? That's different. That's a different thing. Among the patients we have dosed, there are few PD-1 treated failed. For example, PD-1 treated become a disease progression, PD, and but still somehow responded to our treatment. For example, as a stable disease controlled situation. But can we really significantly prove that? Still needs to be seen. But that's definitely is one of the objectives. Yeah.
Excellent. And Jen, I have one more question for you. What information is available on the models in the database?
Yeah, I did share quite a number of different models. So I think the database is a good way of seeing which models are available. And there's actually 4 different databases, one for PDX, one for cell line models, one for murine mouse models, and then organoids. But you can access them all from a single login through any of those one databases. So, for example, for the patient-derived models, you have patient information, clinical information, there's pathology diagnosis. We have over 2,500 PDX in there to search through. In the organoid database is over 600. So there's quite an extensive list to kind of go through, but we've listed them as cancer type. And the goal of having such large databases is to represent diverse cancer types, subtypes, treatment responses, and populations and clinical spread. So that's hopefully quite easy to kind of look at in these databases. And then same with the cell line ones, we've classified them under tumor type. And these models have been characterized for growth. There's response to different agents. There's IHC in there as well. And then you can use a search engine for gene expression levels. So there's whole exome sequencing, RNA sequencing, there's mutation, copy number, fusions, and also there's some proteomics in there as well for some models. And all of these models are QC'd before they go into the database. So they're authenticated by deep sequencing. And actually all this information will really help with that model selection for those preclinical studies. And then through our bioinformatics support team, you can actually sort through the data for responders and non-responders and look at that omics data together with the team to potentially look for sort of predictive biomarkers. So yes, there's a lot of information in there and also the support there to help dissect that data a bit further.
Wonderful. Thank you so much. That's all the time we have for today's webinar. As mentioned, the presentation and the question and answer will be available on demand shortly for repeat viewing or to share with your colleagues. I'd like to take this opportunity to thank our two speakers for their wonderful presentations. Henry and Jen, thank you so much. I'd also like to thank our sponsor, Crown Bioscience, and the wonderful Gen Multimedia Group for hosting this event. Thank you so much to everyone. And many thanks to all of you for watching today. And just a quick reminder before we end the session that on September 13th, we'll be hosting the State of Biotech, which is Gen's flagship virtual event. And this will feature more than 4 hours of dynamic talks and panels, kicking off with a keynote conversation with the 2022 Nobel laureate, Carolyn Bertozzi. Registration is free and there are full details at the Gen website. So for everyone at Gen, thank you so much once again. I'm Uduak Thomas and goodbye for now.